can do it.
Happy dogs. So good. Wonderful. Welcome.
What's a good boy?
What a good boy. What a good girl. Okay. Amazing. Welcome, everyone. How are you? Are we okay? Check the chat.
No. Nobody's saying
Nobody's
anything.
saying,
Happy Monday.
oh, there
That's
we
nice.
go.
Happy Monday to you, too.
Is it Monday? Oh, my goodness. You're right.
We are here.
Okay. That just puts a whole bunch of things into perspective for me. Thank you for reminding me. It's Monday.
Yeah.
Yay. Super good. Okay. Folks are still coming in, so we're not going to, get started. Although today is a very cool day. We're going to kind of take it easy on you a little bit more because things are getting a little bit more challenging on campus and you're probably exhausted. There will be a couple of opportunities for you to stamp and things like this, but mostly we'll just tell you about cool stuff, if that's okay. And then our homework is kind of a way of like integrating the cool stuff. So hopefully that will work for all of you, as well as all of you who are not here, but watching later. And thank you for doing that. We see you.
Hello to you in the future.
Hello.
Hope everything's okay.
Yeah. So just a little note, I think important, the EPP deadline has been further extended. Now, we don't love extending it so last minute because it can be stressful for students like who have already completed it. So we don't usually do this. What we do is what we did before is like well in advance extend the deadline. This is a real sort of emergency deadline extension because of the number of requests that we're getting from students for an extension. And we're going to guess that some students out there are not as empowered to write to us and ask. And so on behalf of them, and thanks to those who did reach out, we are extending the deadline by another 2 days. So it will be on Wednesday at 10 p.m. If you've already handed it in, but you would like to take advantage of those couple of extra days, please do feel free to simply upload a second version and we will mark the most recently uploaded. Okay, so another 2 days for sure. We'll see how this goes. And the reason why we're saying we'll see how this goes is with respect to the other deadlines, the other things, is because unbeknownst to many of us, there's an outbreak on the University of Guelph campus. I am using the word outbreak even though we're not allowed to. We're being told that it is a disease cluster.
Or sickness of whatever variety.
Yeah. So there is an outbreak on campus. It affects you. If you take a look at the wastewater profiles for the university, unfortunately, the last time that they uploaded them was on the 22nd of March, but they were all in the yellow or deeply red zones, which means that there is COVID on campus to quite a large extent. So it isn't a cluster. And so take care of yourselves at the best that you can. And even if you do get COVID, there are things that you can do to help get through it. Rest, right? Don't push yourselves, and hydrate and eat well if you can. Get friends to help you get access to food and all of those things. Okay. So, if you get COVID, terribly sorry. And there are things that you can do to make that a safer and healthier experience. Okay. If you need any help, if you need any resources, please do feel free to get in touch. We are there for you.
Yeah, I think one of the things that you can think about doing is advocating for yourself and for others, even if you are not directly affected by feeling like you having COVID, recovering from COVID, or being worried about COVID, is advocate to your profs if they are planning for an in-person exam. Our college has been instructed to begin thinking about non-in-person exams.
Yeah.
This is a little late in the game because we've been thinking about that since last year, but they've been encouraged. All instructors have been encouraged to do that right now because of these disease clusters of uncertain origin.
One thing that I would like to say, I know this is like totally biased, be kind with your professors a little bit. That this is kind of blindsided all of us. I had to I didn't actually go, I went by Zoom, but I had to go to a conference in Waterloo to find out about this. So where somebody said, I'm really sorry for what's happening on your campus. And I totally faked that I knew because I was embarrassed.
Oh, right.
But then I started like texting and calling folks. Anyway, So they are blind. Your instructors are overwhelmed, overworked, blindsided by all of this, and so definitely advocate, definitely,
Yeah.
and maybe just like a little kinder
Yep.
than what you're feeling right now, because I am freaking furious. And when I ask for something to folks that I know have also been having a hard time, I do try to be kind. It's hard though. Sometimes I totally lose it.
No.
No, I don't. Yes, I do. So I'm speaking from personal experience.
Yeah, but file it under the expect kindness
Yeah.
and deliver it.
Yeah.
Go in delivering it.
Yeah. Okay.
All right.
Just because it's fun. Okay, should we get started?
I just posted one of the Doug in the Backyard videos,
Oh, yeah.
saying we were inspired by this video to repetitively, well, we catch frisbees all the time in the backyard, but...
Okay, cool. We hope you like the video. We love the video.
If it's stilted as you're watching it on ours, take the link later on.
Yeah, and then watch it. Because awesome.
Again and again and again. Even if you're not a dog person, if you're a cat person, that's fine.
It's still a happy video.
It's still a happy video.
What would a cat version of that look like? Okay, serious question.
Yeah, in the chat.
What would the cats be doing
They would be like...
in this video?
They'd be like totally face and cheek wiping. Yeah. Or it'd be
Chasing mice.
Kittens walking off of the countertop into sunbeams.
Zoomies. I like zoomies.
Oh, playing with the laser, of course.
There we go. I like it. That's good.
Yeah.
Meow.
Waiting to pounce on the dog.
Nice.
Glaring at the camera. Oh, I thought that was a suggestion, but I was like joining the chat. I was joining the shot.
Awesome. That's awesome. Wonderful.
Your dog chases lasers. That's super efficient.
Yeah, that's good. Then you don't even need
You'll
a cat.
never lose your frisbee.
Okay, cool. So we're going to tell you two stories, and they are going to be seemingly totally unconnected.
Mm-mm.
But there is a delightful kind of sort of connection.
In the blood.
In the blood. It's my favorite story.
Today's a bloody day.
Yeah. Anyway, here we go. A tale of two hemoglobins.
It was the best of hemoglobin. It was the worst of hemoglobin.
And that's really what
The end.
Well done. That was the whole story. So we're going to tell you about the best, and we're going to tell you about the worst.
You think we're kidding? I've been holding on to that one.
Oh, good one.
Yeah.
Okay. But let's first start with Woolly Mammoth, because awesome.
Yeah, but the way that's titled, we should have like afterwards the bum bum bum.
Okay, Woolly Mammoths, amazing. Wouldn't it be so cool if they were around? I think so. Like in my pocket.
Little pocket-sized ones in the drawer. They all sound like Ray Romano.
No.
Mom.
Okay, so we're going to tell you about woolly mammoths, and we're going to tell you about woolly mammoth hemoglobin. But first, always start with a context, right? About what we know about hemoglobins. What do you know about hemoglobins? Pop it in the chat. Pop it on the slide.
You want hemoglobins or mammoths?
Sorry.
Oh, yes.
What do we know about mammoths? Thank you. Did I say hemoglobin?
You did, but
what
that's all right.
What do we know about woolly mammoths?
That's the name, a teacup mammoth. You just like...
I want a teacup mammoth.
Yes. It could chase around your teacup chihuahua.
Okay, so what do we know?
They were in the movie Ice Age. True.
Lived during the Ice Age. Big, furry, extinct. That captures the whole thing. Well done. Large. Now extinct. Ice Age. What else?
Big.
Herbivores? Question mark? Yes.
Arctic habitats.
Furry.
Interesting.
Maybe related to elephants? A furry elephant. Nice.
Kind of look like elephants.
Yeah.
Warm-blooded, I'm assuming.
Yep. Migrated. Emily, thank you. Yes, definitely migrated, and we will show you that. Cool. Arctic habitat. Nice. Herd living. Yeah. Good. Super.
Little known fact, they made a great flan.
Okay. Yes, all of these things are correct. Anything else in the chat?
Nope.
Warm-blooded. I'm assuming. Yes. Good assumption. Based on evidence. I like it. Okay. Good.
Well done.
Wait. What is this word I've never seen before?
Gomphotherium?
Gomphotherium. We are
Begins to Google.
Google. What is Gomphotherium? Nice. It's
There we
the
go.
genus. Well
Nice.
done.
Using the words.
Good. Love it. The genus. Very good.
From the
Okay.
Latin welded beast.
Amazing. Okay. Admit. Folks are still coming in. Welcome. Okay. Clear. Moving on. So we know some stuff, right? We kind of know the basics, and that's perfect. That's all we really need to know. So here it is kind of in dot form so that you can have it on your slides for later kind of deal. We don't need to go through all of these, but they all are extinct, like you mentioned, which totally sucks. And as recently as about 1700 years ago. So there was definite overlap, obviously, with humans. Human species, as of right now, is about 195,000 years old. And what I mean by right now is that that's the evidence that we have, and it just keeps on crawling back. So maybe we'll go a little bit earlier than that for humans. But definite overlap, and some really interesting research, like super recent, coming out about that population that was on this small island. It turns out that there was a lot of sort of evolutionary decline. So there was lots of evidence of disease, inbreeding, things like this. So take a look for that. Just look for, news articles about mammoths most recently in the last year or two. Cause of extinction, probably a whole whack of bad things piling up on top of each other. So
Oceanic Flight 815.
Really?
I'm just thinking about Lost, like if they were
Oh,
trapped on a small island, they'd be there with the smoke monster and all the gang.
No. Do you know Lost? Have you seen this series, or are we just way too
I think
old?
yes, both things are true. We are way too old, but the pandemic
They
is
do
into
know
its
Lost?
third year, and so everyone's
Binge watching
watched
Lost?
everything.
Okay. If you haven't, it's not bad.
Lost is amazing.
Oh, good.
Even the ending.
No. Okay, cool. Okay, Smith.
In the beginning. So what we're looking at here is a phylogeny. We've been rotating them on, right? So as we go through, of course, we have continued to think about ecology. We will continue. We've continued to think about evolution and have those phylogenies, particularly tree-based thinking, inform how we deal with ecology and now physiology in the tale of the two hemoglobins. So with this lineage, you're looking at time here, left to right. These are epochs, and we are living now with two extant lineages. This digression is really exciting, is we have taught each other for a long time. I was taught, you were taught, up until a couple of years ago, we would have taught you that there were two extant lineages. The Asian elephant seems to be a lineage. The African elephant is multiple lineages. Each one of them kind of habitat specialists.
Yeah.
So
So it would be branched, right?
that's kind of an amazing thing that is collapsed inside of that one. So, but these are the extant lineages, and you can see just kind of nosed out, get the pun, at the finish line is the mammoth. Now the mammoth is different than the mastodon. You can see down at the bottom quite a long time between those nodes would have connected. If we'd been reading our own slides, we would have seen gone through. Yep.
All the way back there.
Look at that. That's
Look at that. Mm.
using the words.
Yeah.
Very good.
Okay, one thing that I notice,
What's that?
check this out. Which group is the mammoth most closely related?
It's not the hummingbird.
No. But which of the extant mammoths? Do you want to give me a stamp on the extant mammoth? Yeah. Cool. And so that to me is talking potentially about this migration, right? Going, Asian elephants are more closely related to mammoths than African elephants are. And so where might they have migrated from? Potentially, maybe. Kind of cool.
No, I think that's kind of great. It's looking at a lineage that probably started in Africa and then left.
Uh-huh.
And then there was some branching.
And then there was some branching.
And then they left.
From that, anyway, you know how to make it all work. Okay, cool. Thanks.
The power of the tree.
Wow.
Now, speaking of left, here we go.
Yeah.
Look at that map.
It's kind of similar to the human one, right?
Yeah.
There's a big difference, though.
No, just I wanted to, because I caused I said a similarity. I don't want to make a confusion here. This is paths not of elephants. These are mammoth. These are genera is the plural of genus. These are genera of mammoth.
Yeah, mammoth.
That we have been tracing through.
Yeah. Good, thank you. Okay, and similar to humans, big differences here, though. Okay, and that has to do with the dates. So take a look. 1.8 million years ago, right in the middle there. Clearly humans were pretty probably not around at that time. Like, I don't think we're going to find any fossil evidence of that anytime
They're humanoids.
soon. They're humanoids, hominids, but certainly not the human species. And
my
so,
red-haired relatives were there.
yeah, it's more about following in the footsteps of mammoths. What the
Big pie plate shaped footsteps.
Which is probably what humans did as they were exploring all sorts of different parts of the globe. But very much after. So, following an established pathway, which is super cool, I think.
And kind of what we're good at.
Yeah. Okay. And this stuff gives me chills because people regularly interacted with mammoths, that people got to see, got to worship, got to wonder at the mammoth, and also got to eat it and benefit from all of the gifts that the mammoth would be able to provide to people, especially
Do you remember where this
at that time.
particular drawing is from?
Yeah, so this is a drawing from Lascaux, which is a cave in France, and the caves in France are just phenomenal. Like the amount of artwork, which often suggests that people at that time, if you have art, it suggests that you had time for art, which is wonderful, and valued art as a means of communication, as a means of community building. And when you engage in artistic practice, you draw what's important to you. And so all of these animals are very, very important. Clearly is the interpretation. I could be totally wrong because I wasn't there.
The interpretation is
but
shared
that's the
by
interpretation.
others. Yeah, those caves are it's a really visceral response. I remember seeing all of these different species drawn there, and then also narratives with hands, probably indicating the number of people, the number of families, those kinds of things.
Yeah.
And I remember being blown away, like crying in this cave because there were hundreds, dozens of right hands, and then a left. I was like,
that's me. So the other thing that we can see in these caves is evidence of mentorship and apprenticeship, which I think is remarkable. So you can see caves. I've been to one, Fond de Gaume. It's closed now to visitors because of the carbon dioxide, damage that can be done. But when it was open, you could see this like beautiful mammoth, and then behind it were also beautiful mammoths, but maybe not as well drawn as that first one, suggesting, and the interpretation was that these were done by apprentices that were in the process of learning how to depict it. And so there's this whole technology associated with it. And of course, you can't just take a look at these with like a spotlight. No. You have to do it with candle or like a flame, because if the flame moves, so do the animals. And that was really the technology that they used at the time to light the caves. And so if you have the opportunity to experience these things, it's truly grounding and delightful. Interesting. If this is of interest to you, I don't know if it is, so forgive me for rambling, but a little bit of trivia. So far, there is only one documented human form that exists in this cave. It is buried well deep, super hard to get to. It's a person, quite clearly a person, but the person has the beak of a bird and a very large erection. And people can't figure out what it means, but there you go. Just a kind of factoid for you.
Yep.
Yeah.
And some in the chat, some people have been.
Oh, good. Yeah, the Chauvet caves are, exactly. Those were the ones that we went to the second. Yeah. Wonderful. Yeah. Cool. Okay. So obviously lots of questions about the extinction, what happened, because damn, they were cool. And certainly it seems to be more and more that it's multiple factors that are kind of piling up and putting pressures on the population. Humans, there is obvious evidence of interaction combined with climate change, though, which would make it more difficult. These things coming together is probably pretty likely that that's what happened. Okay, so
which makes us think about blood.
Which makes us think, no, but one cool aspect about the mammoth.
That's a pun. You'll have to unpack it later, but it is a pun.
Okay.
So I flag it now.
Okay. It's one of those you'll find it funny later thing. Mm-hmm. Okay.
Except I'm finding it funny now.
Okay. So, hemoglobin. Lots and lots of vertebrates have it. Like the overwhelming majority of vertebrates need it. And one of the sort of real challenges associated with being without having a circulatory system, right? So if you're single-celled, is that you can't really get so big that osmosis, breaks down. So circulatory systems and the ways of transporting things within the body is kind of like this revolutionary technology that allowed for things to become bigger in volume. Right? Yeah. Two-dimensionally, something that was single-celled could become quite large if it's just exchanging across its membranes, with the environment. But in terms of like circulating stuff around, you need a system to be able to do that in order to get very big. And so that's just kind of a general thing associated with invertebrate and vertebrate biology is circulation. Now, mm-hmm.
Circulation and different kind of hemoproteins. And one of the cool things, we focus on this one. This is a vertebrate story today, but there are independently at least 4 different kinds of oxygen-carrying proteins that have evolved in animal life independently to solve this problem that you talk about how do you get bigger? Well, you need to carry things around.
That's right.
And this is how we do it. And for us, this is part of the story, first and second, is that this is really how we do it. All of the vertebrates.
Yeah. So basically, hemoglobin is a big pile of protein all folded up in a very specific way, okay? So it's this protein, it's like folded like I don't know, like two ear muffs put together. And it kind of rotates like this. And when it rotates, it either creates the condition of offloading or of uploading an oxygen molecule, okay? So it goes from an oxy to a deoxy state, back to an oxy, back to a deoxy, like this, back and forth. And so obviously in the process of doing that, it is being moved around the body and it's bringing oxygen to the places that need to be there, obviously where the energy is happening, where things are being burned, in order to be able to keep your muscles moving, all of this stuff. So that's basically all you really need to know about hemoglobin. You'll learn way more in your other biology courses, but we need to kind of get that basic. Because one of the things that's really important to note is that the functioning, so the efficiency of hemoglobin is very much related to temperature. And woolly mammoths spent a lot of time standing on very cold surfaces. Now, maybe not ice per se, like floating sea ice, but certainly snow, and certainly like, ice that was on the land. And so they were, covered in fur, but there were areas of their body, and this gets into that whole regional heterothermy thing that we keep on mentioning. There were areas in the body where it was much, much colder. And so usually what happens in endotherms that are in very cold places is there's really the prioritizing of the core to keep the body warm. And sometimes you got to compromise. And so what we often see is that the limbs are much colder. But you need your limbs to walk or run. But hemoglobin isn't as efficient when it's colder. But those limbs are colder because you're preserving the core temperature. So
how are you going to balance that?
It's kind of a problem.
They're trying to turn over their car.
Yeah. Okay. So some folks had some questions about these things, and especially at a time where, genome sequencing was kind of a new thing. Is that fair?
Yep.
It was like a thing. Yep. They were like, can we look at this? This isn't a fossilized.
Nope.
This is just a mummified. This is like real tissue, dried. It's brilliant. And so there's tons and tons of DNA, and DNA is an absolutely fascinating technology. It is by far the best way that we know of to store information, if you think about it. Like the CD. Do you remember the CD?
No.
No. And like you'd scratch it and that would be the end of it. A USB key, like you pass it by a magnet and it's gone. You know, all of our ways of storing information pretty much suck compared to DNA. DNA lasts tens and thousands of years, if not more. So it's an amazing technology. And people are actually like writing text using 4 base pair combinations, just like in a binary type of a code. It's kind of cool, and will be around for a long time. So some folks thought this whole hemoglobin thing might be a really cool way to explore the mammoth genome and to understand its adaptations a little bit and to kind of reconstruct, right? The whole idea about reconstructing a mammoth and then studying it would be really difficult. So instead, let's just pick parts of the mammoth genome to study and see what we can learn about it. Kind of makes sense, right? Okay. So what they did was they found some mRNA. They took a look at the functional protein for hemoglobin. They sequenced it and they started to ask some questions about it relative to the vertebrate sequences of known, of other sort of extant species.
So some of this work was done at the University of Winnipeg, very cold place. Kevin Campbell and a bunch of friends. And what they found as they started to rebuild these mammoth, parts of a mammoth, essentially, as much as a part of rebuilding a tusk or rebuilding some hairy bits, some hairy hide, was similarity. You remember the similarity, that node that you identified, where had mammoths branched off of? Well, what they found here was real similarity in between the conformations of the woolly mammoth hemoglobin. It was most closely related or similar, most similar to the Asian elephant and less similar to the African elephant, just as the phylogenetic kind of history would make you think. This doesn't always have to be. Genes and species can evolve at different rates, and we will share a lot. So this was kind of interesting, and it suggested that this is something that is likely under selection and potentially associated with the speciation of these lineages.
Cool. So we're going to take a look. Essentially, we're kind of exploring a new level of organization that we haven't yet really explored in 1070.
Say something to Doug.
Yeah, sure. Our dog is barking. It's really loud, so we're gonna.
Perhaps you can hear it.
Yeah. Okay. So new level of organization that we're introducing to you, and that is the molecular level, right? These are molecules, biological molecules that that have adapted potentially, or that have at least function, biological function. Okay. So, hypothesis, and please feel free to share on the screen or in the chat. Woolly mammoth hemoglobin had properties that allowed it to function in extreme cold, right? It's not like they wore socks. So somehow their hemoglobin was able to function despite the fact that it was just really cold in their feet. So what might we then predict about hemoglobin? What could a prediction be that would help us investigate how that function was achieved? I'm just going to pull up the chat here so I can read it too. Oh, there it is. Cool. Anything that we can maybe predict about hemoglobin? Supercooling. Good, good, good. What else?
Antifreeze proteins, question mark?
Nice. Like, so does hemoglobin have like antifreeze proteins around it potentially?
Mm-hmm.
Let's just imagine, I see where you're going with this, and that's totally fine. Let's just imagine that it wasn't that cold in the mammoth appendages. So let's imagine that it was like only 2 or 3 degrees Celsius difference, that the core body was like 37, but its limbs were like 34. right? So we're not really talking about the area of freeze tolerance or intolerance, but just what might we look at with hemoglobin? What might we predict about hemoglobin?
Less rigidity at lower temperatures.
Yeah,
I like it.
there we go. There we go.
The
Yeah,
shape
yeah, yeah.
might generate kinetic energy
Super.
in the blood.
Super, super, super. Yeah, yeah. I like the idea of supercooling because it does suggest that, like, for some molecules, there are these differences in terms of range and tolerance, and that this one therefore had to be able to operate in colder. That's cool, right?
Mm-hmm.
I like the integration of that. Cool. Nice. Okay, so yes, let's take a look at the actual molecule, right? The actual, protein sequence, right? So one thing we could say, and again, we can say so many things. We're just going with this one to kind of illustrate, right? Is that the hemoglobin structure will be more similar to that of other Arctic mammals than to the hemoglobin of related tropical elephants, right? That there's something going on in the Arctic that if you can't kind of figure out a way to adapt to it, you've got to get out of there, right? Or else your oxygen won't offload. So something about the hemoglobin is similar with other Arctic species compared to its closely related species of other tropical elephants. So they did an experiment, exactly this. They looked at the sequence of the hemoglobin compared to other extant species and also looking at other extant Arctic species. And they looked at the ability of that hemoglobin to basically bind over a wide range of different temperatures, okay? So if we go with that, and we don't only have to go with that, but let's just go with that. It turns out that mammoth hemoglobin was more efficient at colder temperatures compared to other elephant hemoglobin. So there was some kind of difference there. And it turns out that, yeah, mammoth hemoglobin did use the similar mechanisms as other Arctic mammals, okay, like the fox or the muskox or the reindeer. Okay, so their hemoglobin was more similar than the hemoglobin to tropical elephants. Do you think this similarity is the result of shared ancestry? So now should we go and like redo the phylogeny, or is this a case of convergent evolution? Which one do you think it is?
Yep.
Well
done.
Super
Well
good.
done.
Yeah. You've got one molecule, a fundamentally important molecule, like critical to life. So if there's this real sort of external pressure, right, then it's like an environmental restriction, right, limitation. Then it's not surprising that you'll see similar ways of adapting to be able to survive and thrive within that context, right? So it's not super surprising. Don't rewrite the history books and say that, foxes are actually more closely related to mammoths than mammoths are to elephants. That's not like, kind of logical, right? But an important thing is to recognize where convergent evolution is likely to happen. So well done. Okay, so woolly mammoth, the big kind of take home here is that the woolly mammoth hemoglobin can offload oxygen in colder temperatures than just regular or than tropical elephant hemoglobin.
So another Arctic occurrence.
Wonderful.
But one that we've resurrected past an extinction barrier, which
Yeah.
is kind of cool.
So cool.
So one last story about mammoths. And then we leave because we have to talk about fishes. And that's this, and you've probably heard about this. This has been in the news all over the place. And this goal, we talked kind of and laughed about creating a teacup mammoth. But there is a literal and well-funded goal now with infrastructure and backing and science. And their goal is to create essentially a hybrid between an Asian elephant and as much mammoth DNA as we can using CRISPR. The reasons they give in their company website, in their manifesto, in their mission statement is because they are making links to how the tundra in the north, in the permafrost and across the steppe or the boreal, would be more easily frozen when it was trampled. And because it's not being trampled any longer, because we no longer have mammoths, it's melting faster and it's giving up its stored carbon. And so they're saying that this is not just an ivory tower, kind of like, let's bring it back and see what happens, like a Jurassic Park kind of a thing. It's like, no, let's combat climate change by having mammoths stamp the steppe.
Amazing.
That seems like a stretch to me, but I'm wondering what you think. So put it in the chat. Or
vote.
Yeah, vote.
What do you think?
De-extinction's a great idea. Bring it on.
Yeah.
Barbecues. Or, in a world of limited resources, are there other things that we ought to be doing with the funds that we might, some on the border. So I would try and convince you. There are other habitats that have not yet gone extinct. They contain species that are not yet extinct, but they will be by the time your kids or your family takes this class again in the future, or by the time you teach this course in the future. And so I think of it as like, yeah, that's an interesting hypothesis, but let's direct some resources towards things.
And I agree with you. A counter argument, though, might be all of the things that we will learn from engaging in this process. And if it has to be done
the moonshot, yeah.
So if it has to be done on mammoths, because folks will give money to mammoths, so be it. Who cares, right? It's a complex organism, and if we can resurrect it, think about what we might actually do for.
Yep, and I tend to agree with that because lots of times people throw up the idea just the strict dichotomy of saying, oh, there's this money, we should be doing it with something else. It's like, there's lots of things that don't raise money. I happen to work with all of them. And so there isn't a pool to protect the extant staphyliniid of Costa Rica. That pool does not exist, but there are people who would fund this.
Oh, yeah.
So now what we need to do is fool them.
Fool them. Okay.
Into thinking about fishes.
15
minutes of fishes. Go.
Okay. Ice fish. Fantastic creatures.
This is what they look like.
That's
first off. That's like bananas.
Just amazing. You can see through them. You do not need an x-ray.
Yeah.
You can see all of their parts. It's like a teaching model of excellence, right? So no, they're fantastic. Maybe you've never heard of them. Maybe you have. If you haven't, yay, today is the day that you are going to hear about ice fish. They are phenomenal. They are Antarctic fish, and they are endemic to the Antarctic, to the Southern Ocean. You will not find them anywhere else. There are 16 species that are currently recognized. I wouldn't be surprised if there are a whole bunch more. They are considered to be a dominant fish taxon within the Southern Ocean. So all the way around Antarctica, you will find ice fish, and they are there in totally respectable numbers. They live in water that is between the temperature of -2 to +2, and that's pretty much it.
A frosty bath.
That's right. That's what they like. Ice fish. Here's another one. It's getting a little milky, a little creamy here on the side there. As they get a little bit older, they get a little less transparent, but still a totally beautiful fish and fancy. The one thing that is particularly freaky about them is that they have no hemoglobin.
That's bananas. That's a science paper in 1950 going, what?
Okay, so it's freaky. It's a vertebrate with no hemoglobin. This is like huge
So
news.
It should be a flat sheet.
That's right, like just belly up, really.
Yeah, it should not work.
No functional red blood cells to speak of. It's been completely lost in its genome. It's not there anymore. It's not like it was just shut off no, it's not there. As a result, in order to live, there are some things about the ice fish, like they have massive hearts, okay? They also have lots and lots of blood, and their blood looks like the vial here on the left-hand side in image A. This is ice fish blood. This is other fish blood. There's a big difference there. It is remarkable to me, okay? Yeah. They have a really costly circulation system, so it takes a lot of energy in order to be able to get things moving around their body, okay? Good. So how? Let's go back to some of the evolutionary story, because I think it's kind of really cool. So we talked about antifreeze proteins. We talked about all sorts of other adaptations to being able to survive in really cold water. Here, what we've got is a clade of cold adapted fish. Here's the Antarctic clade right here, right? Starting with the notothenoids here. They're the first to branch off. Right here, we see the evolution of antifreeze proteins. And so all of these can live in not just cold water, but like damn cold water, right? Like super cold water. That's a biological term.
That's an SI term.
Okay, so we also have the secondary loss of antifreeze proteins in some of them. So maybe some of them didn't, decided, nope, not going to live in damn cold water, just going to live in cold water. But anyway, here is where I would like you to focus your attention, because what we have in this one group of the ice fishes, we have the loss of hemoglobin. Is there anything else that you want to draw folks' attention to this?
No, not
No?
yet.
Okay, good. I think it's really cool right here. And then if you sequence these things to try to find out, remember I said like the gene is gone? The gene is gone, like really damaged.
Yeah, it's really gone. So there's a big chunk of it at the top, and what you're looking at, these colored kind of popsicles down on the left-hand side of the screen, are different lineages that have different amounts of this kind of gene left over. So our out group, we've got at the top with some functional bits. But then as you see, as you get further and further in, immediately there's a loss. Interestingly, if you look in the center at the annona, there's a different, so this suggests perhaps
multiple losses.
Ha!
Look at me.
Yeah, you just read the tree.
Yay! Okay, yeah. So gone, disappeared, no hemoglobin.
Bananas.
You couldn't even get it back, right? Like it's just not there. You'd have to start from square one all over again. So how is it that we have vertebrates without hemoglobin, right? No hemoglobin, no red blood cells. The gene is lost. This is definitely a WTIF. What the ice fish? Kind of a moment, right? I can make the dad jokes too, see?
That's very good.
Okay, so how does this work? And for the longest time, people got really excited about this because it was this adaptation. Something was going on, and this was like a super evolved fish, right? That's what people thought. So we get all sorts of studies. I hope you can appreciate some of the sort of medical applications of some of this research. So folks were really thinking, about people with blood diseases of many, many sorts. How can, the ice fish help us understand a little bit more about being able to survive, maybe not without hemoglobin, but with less hemoglobin? There was all sorts of money put into this, including a good friend of mine who spent a lot of time in the Southern Ocean in Antarctica studying ice fish because of the potential for these adaptive qualities. But maybe there's an alternative, right? And we tend to always drift towards adaptation, right? If something exists, it's because it's adaptive. We totally forget about all of that stuff that we talked about in the evolution unit on these random mechanisms, right? So if you remember, it's the non-random mechanisms that lead to adaptation through natural selection. But what about gene flow? What about founder effect? What about bottleneck? What about just genetic drift generally, right? These things are part of evolution, but they yield non-adaptive strategies. This very well could be, and actually it kind of is, I'll like ruin the punchline, this is a case of non-adaptive evolution that is facilitated by an absolutely wonderful environment, okay? So, evolutionary mechanism, and we'll just skip the brainstorming thing because we want to wrap up on time. So, if hemoglobin is non-adaptive, if hemoglobin loss is not adaptive, it's super costly, right? You need to have a massive heart. You need to have big blood vessels. You need to be pumping tons of blood because this is now all by osmosis, right? Like this is not anything other than how bacteria breathe. What are the mechanisms? So gene flow, genetic drift, bottleneck, those types of things start to come into play. Okay, we can get rid of gene flow for the most part and just focus on genetic drift. And so somebody figured it out: when bad things happen to good fish.
Great paper title.
So good, right? They started to ask about, does it really confer an advantage? In some ways, it is clearly more costly, right? And that was a wonderful way of thinking about it. Instead of mutation that destroys the hemoglobin function, but was not fatal due to very high oxygen environment. So this is where we get into understanding how the environment can actually facilitate the evolution of maladaptive traits.
Apparently maladaptive traits. Yeah.
Right. So it's called environmental facilitation. When we talked about the mammoth, we were talking about environmental restriction, right? It was imposing a limitation on the hemoglobin to be able to offload oxygen, so adaptation evolved. Now we're talking about environmental facilitation, and in this particular case, the thing that facilitated it was the temperature of the water itself. So cold water can hold more oxygen than warm water. And if that isn't immediately obvious to you, think a little bit about when you put a pot of water on a stove. You put cold water in a pot on the stove and it begins to heat up, right? What starts to happen? The energy starts to rise. You may even have it rock a little bit if you don't have the flattest of pot bottoms, right? It starts to rise a little bit. Then you start to see bubbles, right? The release of gas. Okay, so what's being released there is the dissolved gases that were in the water. So you're deoxygenating your water by heating it up. The molecules of water are occupying more space. It's pushing shit out. Okay, so that's the good stuff, right? The oxygen. Cold water can hold a lot more oxygen. So in the context of this hyperoxygenated environment, losing your hemoglobin, it was probably hard for the first few fish, but it wasn't a massive crisis, okay? They were allowed to continue to be able to survive and thrive because their environment had so much oxygen.
And they must have done better on some level because they weren't investing in this costly protein. And right. This is the same if the water and the oxygen thing is still, you're still thinking about it. Think about it tomorrow morning as you're making your coffee or your tea, because that's why you're using, or why you're going to get better tasting coffee and tea with cold water. It's because the oxygen that's still in there, the greater amount comparatively.
So, I love that question there. You're bang on with where I'm going with this. What will happen if the water becomes warmer? Oh no.
Oh no is right.
Oh no is perfect. Yeah.
Yeah.
Okay. So, here's something to kind of read if you want to kind of practice what we would love for you to be able to do, what we're kind of in terms of for the next topic test, is to be able to read an abstract like this and go, oh yeah, that's gene flow, or they're talking about bottleneck, without actually seeing those words. So try to connect those concepts to the biological story that's happening. Okay, so you can use that to practice. We've kind of underlined some key bits that should hopefully kind of either cause you to Google stuff or cause you to make connections to things you already know. And here's a question that you can use in order to be able to test your knowledge. And we'll take it up next time. But in order to even help, it might be helpful to first start with this table. This table kind of goes through the mammoth and the icefish stories and asks you questions. Super simple. It starts out. Okay, so don't overthink these. You know these.
Yeah.
But then, as we get to the bottom, we're going to try to help you understand the differences between the mammoth and the icefish story and where they are different. So where are they similar? Where are they different?
So so we're at the end of time, right up flush against it. But there's one more slide, and it's to make me remind ourselves to tell you that these stories, there's a lot of these two stories. There's a lot of literature. There's pop culture, there's whatever. And this is one lecture in particular. They all have it, but the PDFs, when you get them, are hot links. So there's a link to a chapter in this particular book about the story behind the understanding of the icefish. And there's CRISPR. Anyhow, you can go through, and if this has been kind of interesting and fun, there's other reading that's behind there.
Cool.
So we circle back to the start and say there's lots of stuff going on campus, so take care of yourself and be kind as you request that which you are deserving of.